T-bending of chromosomes. T-bending is actually for the telomeres. Telomeres are the ends of the chromosome. When we stain ends of the chromosome, the bending pattern is called as T-bending. In case of T-bending, that usually identifies subsets of R bands which are present at the telomeres of the chromosomes. Here we can see in this diagram that the ends of the chromosome, which are called as telomeres, this is the centromere of the chromosome, C for centromere, this is P arm, this is Q arm, and these are the ends which are called as telomeres. When we stain ends of the chromosomes, which are called as telomeres, the bending pattern is called as T-bending. Here we can see that ends of the chromosomes, they have been stained. So this is the example of the T-bending. Procedure how to perform T-bending of the chromosomes. Once the age of the slides is 7 days, then the slides, they are placed in a solution. The name of the solution is PBS for 20 to 60 minutes at 87 degrees centigrade. Then arrange the slides with the help of same solution, that is PBS solution. Stain with 3% GEN731 in phosphate buffer at 87 degrees centigrade and leave the slides for 5 to 30 minutes. And then the slides, they are arranged again. Once slides are arranged again, then the slides, they are stained with a chemical, that is hoist 33258, that is a specific designation of that stain. Keep the slides for 10 minutes in that particular stain. Then arrange the slides with phosphate buffer and examine the slides under fluorescent microscope. When we perform all this procedure on the chromosomes or the slides which contain the chromosomes, we can see the telomeres of the chromosomes have been stained and they are showing the dark bands at the ends of the chromosomes. In case of T-bending, there is alternative procedure by which we can stain the slides. That is, the stained slides, they are covered with the cover slip and placed in a wet chamber under UV lamp for 2 to 3 hours, or the slides can be placed directly under the sunlight for 2 hours. So this is an alternative procedure by which we can stain the telomeres of the chromosomes. Remove the cover slip and stain the chromosomes or the slides with GEN731 for 10 minutes. Arrange the buffer, dry, and mount the slides in DPX. So this is the alternative method by which we can stain the telomeres of the chromosomes, either by putting the slides in wet chamber under UV lamp, or we can also put the slides directly under the sunlight. T-bending of the chromosome is the bending pattern in which we identify or we stain the telomeres of the chromosomes.